Okay. Well, I hope everybody's doing well. I don't know. You can tell we've been allowed to come back to the school. Professors on a very limited basis have been allowed to come back to their office because a lot of people, because we had to leave so quickly, we didn't get a chance to get everything. So I'm recording this from my office because I want to, my house has too many distractions. I want to give you this quick, relatively, review for the final. Now the final will be open Wednesday morning, and it'll be open until Thursday night. So 2 days to take it. I'm in the middle of making it. I'm hoping to have 100 questions. It will be an hour and 45 minutes to take it. It's a little over a minute a question. It shouldn't take that long. Got to make sure it's, I think the tests have been pretty fair. From the grades that people are getting, it seems that it has, because most everybody is doing better than they usually do. So I think that I'm excited about that in one way, but I want to make sure that people aren't doing it the wrong way. So anyway, that's the plan as of now. It's not official yet because the dates are, I haven't finished the test. I got to go through each chapter and figure out, about 10 questions, 8 to 10 questions from each chapter to put into it. Now, before we get to this little review, I want to make sure that you know that it is a comprehensive final. It is not droppable. It is absolutely not droppable. It's 15% of your grade no matter what. And just to put your mind at ease, a lot of people get upset about these comprehensive finals because the first thing they think of is, well, that's not that's going to be so hard. There's so much material. Hopefully all you got to do is review on it, because if I'm only picking about 8 questions, there's 12 chapters. The last chapter, again, we'll talk about as we get there. It is the special senses. It's really going to be about the eye and the ear, a few little receptor things that you had to go over in lab. That's basically going to be it. I'm not going to take a ton of questions on that. But if we're looking at it and we divide it up evenly, that'd be about 8 questions per chapter. Now, there's some chapters that are going to have a little bit more and some that are going to have a little bit less, but that's the average. If I'm only asking 8 questions per chapter, that means that I can only get the highlights of it. So don't get overwhelmed in digging too deep. Make sure you know the basics of the highlights. So as I go over them, that's what I'm going to be hitting on, is the highlights of this. Right? This is what I would usually do with my classes if we were able to meet face-to-face for this. My last class was always doing a review for the final. So don't let it get you. Don't get too wrapped up or too concerned or too anxious or nervous about it being a comprehensive final. It is the highlights. And, it's going to be the simple things, and I won't say simple, I shouldn't say simple. It's the highlights of things that you have to know as you move forward. If you don't know some of these things, it becomes a problem as you move forward anyway. So most of this, you shouldn't have to study a whole lot. It should be really just brushing up on things. Now, I'm going to kind of switch. I'm trying to use the Zoom ability to have this. I'm trying to figure out. Now, I'll tell you, I'm not really the best with the Zoom. All right, so I'm just putting this up. Hopefully you can see it. This is the PDF file I sent out with all of the it's really the learning outcomes that are on your syllabus, or the document formerly known as the syllabus, that I just went through and highlighted certain things. So like, for instance, on this, you should know that, it's supposed to highlight. I don't know what happened there. Why is it not cooperating with me? Defining anatomy and physiology and how they're related. That's super important. We're studying a class called Anatomy and Physiology. You should be able to tell me what anatomy and physiology is, right? And so just kind of trying to make sure we're all on the same page here. And I'm trying to work this because this isn't something I'm very used to. So again, let me make sure I'm still sharing the screen. All right. Sorry, hope everybody understands. I'm learning this too. So anatomy and physiology is explaining how they're related. So you need to know what anatomy is. Anatomy is the study of structure. My hand is built like this. Physiology is how it works. It's designed to be able to move and grab things. They are intimately intertwined. What something looks like is going to be determined by what it does, and what it does is determined by what it looks like. So again, they go literally hand in glove, so to speak, and how it works, but you have to know that. Metabolism. Metabolism is a very important concept to understand. Metabolism is simply all of the chemical processes in our body. Now, a little later on, we're going to, we'll talk about it, but we talked about the two types of metabolism. They've got things that build things up and things that break things down. Anything that's building up is called anabolism. We talked about that. Anabolic means to build up. Catabolism means to break down. So anything that's breaking stuff down is catabolic. So metabolism is the sum of all those things in our body, and our body's going through both of them. As we eat our breakfast and it gets into our intestinal tract, it starts getting broken down. That is catabolism. Our body's going to absorb it. It's going to go out to our body, to the different tissues, and be used to repair things. It will build it back up. That's called anabolism. Now, again, here we explain the importance of homeostasis. Homeostasis is a pretty big word. It's the balancing act. And the importance of homeostasis is this is how our body responds to outside influences. Homeostasis is the sign of life. If I can respond, a rock doesn't respond to things, right? So homeostasis is how our body responds to things and keeps things normal. Now, through this class, we've talked about a couple of really specific ones. Calcium, for instance. We talked about calcium balance, how our body regulates blood calcium. It's homeostasis, and it is done most always in negative feedback cycles. All negative feedback is if something moves away from normal, the body will do something to pull it back into normal. That is homeostasis. So another thing that we talked about in this chapter is something that you went over again also in your lab. Identify major body cavities. Right? So I do expect you to know the three areas that we looked at, those serous membranes, the heart, the lungs, and the intestinal area, the abdominal pelvic area, I'll just say. The heart is called pericardium, the lungs are called pleura, and the abdominal area is called peritoneum. The two membranes, the one that's on the organ is called visceral, because viscera means organ. The one that lines the cavity is called parietal, and parietal means outer shell. And so if we look at those, you should be able to tell me that the membrane that is on the heart is called the visceral pericardium. The one that lines the cavity is called the parietal pericardium. Now, those are basic. You went over them multiple times in lecture and multiple times in lab, so I'm not going to go over all three of them, but you should know those. Now, chapter 2 is always the one that gives people, fits. Defining chemistry and how it relates to anatomy and physiology is a pretty big thing. Defining chemistry is simple. Chemistry is the study of matter, and matter is everything that takes up space and has mass, which means all of the periodic table, all things that are made up of atoms are called are material, and that's what chemistry is studying. And how it relates to anatomy and physiology, first, the anatomy of something is the building blocks of what it looks like. The chemistry of bones, where it's calcium phosphate and organized into these osteons and grouped together to make bones or whatever, that's anatomy. The physiology is how the chemistry works. Look, physiology is super important in our body. Again, how our body regulates the calcium levels, goes to physiology. What is calcium used for? As calcium's the end entry thing into an axon and the axonal bulb at the synapse to release the neurotransmitter. And so the chemistry of it is pretty important. Identify the 3 types of chemical reactions. Again, we talked about these. The 3 types of chemical reactions are, we have decomposition reactions. When we have a larger molecule that gets broken down, we call that catabolism. We have synthesis reactions, which we have smaller molecules that bind together to make larger ones. That is called an anabolic reaction. And then we looked at that weird one, which we just called an exchange reaction, where we have 2 molecules, let's say AB, and it gets in the presence of CD, and they intertwine. And then I have A and D and B and C, or however you want to mix it up. It's an exchange reaction. The next thing on the list here is about pH. Now, pH is a very tough thing. Again, I put here, you know, basic understanding. pH is the measurement of hydrogen. You have to know that, right? Now, in our body, we have a pH range that is set by homeostasis, right? 7.4. Now, the understanding basic mechanisms of pH is that pH, again, is a measurement of hydrogen ions, and it goes from 0 to 14. 7 is the middle. And it's a measurement, but on one side of 7, from 0 to 7 is acidic, and then we have extra hydrogen. At 7, we have perfect water. For every 1 hydrogen molecule, I have an OH molecule that binds with it. So I have H2O. Now, from 7 to 14, I have a deficiency of hydrogen ions. That, and said another way, is I have excess hydroxyl, or OH. Now, in our body, 7 to 14, which in chemistry is called basic, in our body it is called alkaline. And our body's pH, which is called physiological pH, is alkaline. It is 7.4, so it's a little bit on the alkaline side of the pH scale. There's a range, 1 point, right? So 7.4 is the middle. So 7.35 is on one side, 7.45 is on the other. All right. Now, organelle. Now, this again is a really big deal. Organic molecules, right? So the organic molecules, and I'm going to put a little light on here. I don't know. I'm feeling like I'm all in the dark here. I don't know if that helps or hurts, and I think it kind of hurts. All right. Organic molecules. So organic molecules, there are 4 organic molecules. 3 of them, all 4 of them are super important, but 3 of them I'm definitely going to want you to know for sure, the building blocks of. But we have carbohydrates, which the building blocks are called monosaccharides, simple sugars. The main simple sugar is glucose. Then we have proteins. The building blocks of proteins are called amino acids. Proteins are long chains of amino acids. And they have a unique structure. Now, things like enzymes are proteins. That unique structure is a really important thing because the unique structure is what allows for enzymes to work. And we'll talk about enzymes at another time. And then lipids. Lipids are fats. And lipids are the main type of lipid that we see is called a triglyceride. In our triglycerides, the main building block is going to be 2 things. We have what we call fatty acids and then a glycerol backbone. The fatty acids are going to be one of 3 types, or one of 2 types, excuse me. They're either going to be saturated or unsaturated. Saturated means there's a hydrogen at every level. Unsaturated means there's not. And saturated fatty acids are generally animal in origin and will be solid at room temperature in general. And then unsaturated are going to be liquid at room temperature and in general are what we call plant fats, like vegetable oil. All right. And then the last one is nucleic acids, which are obviously vitally important because that is DNA and RNA. In nucleic acids, the building blocks are nucleotides. And although I just said the big 3, the first ones, you do need to know about nucleic acids, about the building blocks being nucleotides. In cells, now I want to go back, and I guess I need to kind of, this one isn't starred. And I want to kind of just briefly talk about the way atoms interact with each other. All right, ions are vitally important. Right, so an ion is an atom that is either gained or lost an electron. If it is lost an electron, that means that it has gotten smaller, and smaller is catabolism. So it's a cation. If it's lost an electron, it is, since electrons are negative in charge, that means if it's lost an electron, it is positive overall. If it has gained an electron, since electrons are negative, that means it's negative overall, but since I've gained an electron, I've gotten larger. So an anion is a negative ion. And so we have two main types of bonding: ionic bonds, which is how we find out about pH. All right, ionic bonds are where two ions are magnetically attracted to each other. It's an ionic bond. And if one of those ions is a hydrogen, then it's an acid. If one of the ions is a hydroxyl group, an OH, it is a base, or in our body, alkaline. If it's not, if it's neither one of those, it's simply classified as a salt. The other type is called a covalent bond. And if you remember, co means to share. Valent is talking about outermost electrons. Valence shell electrons, or valence electrons, are the outermost electrons. And so if I have two atoms that share an outermost electron, that's a covalent bond. And again, you can, it's not going to be on the test, but I'm telling you, I think that this helps you understand a lot of what goes on if you get this. If the two atoms share an electron evenly, it is called a nonpolar covalent bond. And nonpolar covalent bonds are lipids in general. If they don't share evenly, then one side will have the electron more than the other, causing the side that has the electron to have it longer become a negative in charge and the other one positive in charge. So it forms these poles. So they're called polar covalent bonds. Polar covalent bonds are what we call water soluble. That's why water and oil do not mix, is because the polar covalent bonds, the ones with these little poles on either side of it, have nothing to grab onto for the lipids. So that causes them to pool together. Now, chapter 3 with the cells. This is probably going to be one of those that I'm not going to have as many questions in, because I want to have, again, the nervous system is going to have several questions, it's going to have more questions in it. So chapter 3, the outlining the structures of a composite cell. Remember, the composite cell was that little fake thing that, it basically cells are going to have a cell membrane, a nucleus, and cytoplasm. Remember, cytoplasm is not simply the liquid in a cell. It is the liquid plus the organelles. If we're talking simply about the liquid inside a cell, it is called cytosol. All right. So the organelles, briefly about the organelles, the big ones, mitochondria, you definitely need to know the mitochondria is the powerhouse of the cell. Ribosomes are where proteins are made. That's a manufacturing plant for proteins. And again, we'll talk a little bit about proteins here in a second. I'm not worried about moving in and out of the cell, organizing the cell cycle, and explain how it divides. So this is about mitosis, right? So mitosis is a 4-part process. The cell cycle is 5 parts, because we have normal cell life, which is called interphase. Mitosis is all about the dividing of the genetic material. And there's 4 parts to it: prophase, metaphase, anaphase, telophase. If you put it all together, I passed my anatomy test. And so in prophase, this is when everything starts being undone. The DNA material, which is called chromatin, starts coalescing together to form what we know as chromosomes, those X's. The nuclear membrane starts dissolving, and then as those chromosomes get to the middle along the equator, which we call metaphase. It's a brief period of time, and then they get pulled from side to side. That's called anaphase. Now, during anaphase is when the whole cell starts dividing, which is called cytokinesis. And then once that goes on pretty quickly, things start repairing. The cell membrane the nuclear membrane starts reforming. The chromosomes start unwinding to chromatin. At that point, then it is telophase. All right. Now, in cellular metabolism, this is a pretty big one. We talk about this in 211 as well. Anabolism and catabolism, we talked about those earlier. I'm not going to go back into it, but anabolism is building up, catabolism is breaking down. Cellular respiration, this is a very important thing. Understand, cellular respiration is a three-part process, right, to get it done well. There's 3 steps and there's 2 stages, all right? The stages are simply referred to by whether oxygen is needed or it's not. The first stage does not need oxygen, so it is called anaerobic. The second stage needs oxygen to happen, and so it is called aerobic. The second stage has 2 parts to it. So when we look at it, it's talking about taking glucose into the cell and breaking it down. So if I bring glucose into the cell, the first thing that happens is it gets broken in half. Now, the word lysis means to break down. And so glycolysis is breaking glucose in half. And this is going to happen with or without oxygen. So this is called anaerobic. Anaerobic, the first stage, has 1 step to it, and it's called glycolysis. If oxygen's present, those 2 little molecules that are formed when glucose is split, get affected by the oxygen and get brought into the mitochondria. Now we're into the aerobic stage. And the aerobic stage has 2 parts to it. The first part is called the citric acid cycle, or the Krebs cycle. That is it's like a chop shop. It takes those molecules and starts breaking them down, pulling parts out. And then the third part, right, we're still in aerobic respiration, is called the electron transport chain. Now, the aerobic respiration is going to produce a lot of that molecule called ATP. ATP is the molecule that our body uses as energy. And so as long as we can use oxygen efficiently, we can produce a lot of ATP. And so that's how cellular respiration works. Again, we go over this in 211 again, and a lot of students get tripped up because they don't learn it here. They memorize it perhaps, but they don't learn it. You should learn this. Glucose comes in, it gets broken in half. Glycolysis happens with or without oxygen, so it is called anaerobic. If oxygen is present, it gets brought into the mitochondria, after it mixes with the oxygen, it's called aerobic. The first part of it is called the Krebs or citric acid cycle. The last part of it is called the electron transport chain. 3 steps in 2 phases. Now, the process of protein synthesis, right? Protein is made. We just talked about how protein is made in the ribosomes, right? So understand that ribosomes are where proteins is made. Now, the recipe for it is in the DNA inside the nucleus. And so we have a process that's going to copy down the recipe from the DNA and take it to the ribosome to create the protein. Now, the process that happens inside the nucleus is called transcription. So I have a molecule called mRNA, messenger RNA, that goes and makes a copy called transcription. Transcription means to write down, literally. And so it transcribes the recipe. That messenger RNA leaves the nucleus, goes out to find a free ribosome, and in the ribosome, it'll start being pulled through little by little, and then these things called transfer RNA come in and look at the 3 parts because it's all written in groups of 3 inside the DNA. It's called triplets. The messenger RNA is called codons. The transfer RNA is called anticodons. But each one refers to an amino acid. So the transfer RNA brings the amino acid and puts it in place. As that happens, that is called translation. It is taking the written copy that the messenger RNA did and wrote down, and taking it and translating it into a useful structure, which is the protein. So that is the way it works. Now, again, I can't spend too much time on it. I'm trying not to I'm trying not to make this a 3-hour video. Tissues. All right, so now this is another one that I'm going to tell you is not going to be a huge part of this test. This is, like I said, if I average it out, there's 8 questions per chapter. There's a few chapters that need more than 8, so there's a couple chapters that we'll have to sacrifice. I think tissues is one of those that I won't ask about and won't ask a full 8 questions. All of the tissue questions will be pretty much straight from lab as well. All right, so we're going to have first, well, and I say that straight from lab, and the first one isn't really a lab thing. You know, I'm not saying I'm going to use these as questions, but you should know the difference between a desmosome, the gap junctions, you know, the junctions between them. The gap junctions are going to be the tubes between the cells. The desmosomes are the welded spots. And then we had the tight junctions, which were the sewn stitching that was waterproof. Desmosomes, if I ask anything, it's where it's a welded or very strong bond between cells. Gap junctions will always be about tubes that are connecting, allowing the cytoplasm to go back and forth. And then the tight junctions will always be considered a waterproof barrier. All right, so, but this is the thing, it says 4 here, but I said the 2 major types of tissues on this: epithelium and connective tissue, and general characteristics. Now, in here, again, no pictures, but you should know a little bit about the basics of the epithelial and connective tissues. And I'll briefly go over them. Epithelial tissue, remember, the naming is important. Simple means 1 cell layer thick. Stratified means multiple layers thick. Then we have the shape. Squamous is flat, cuboidal is cube-like, columnar is column-like. Right? Then we have the ones that don't fit into anything. Transitional means it changes shape. Pseudo-stratified means that it looks like it's multi-layered, but it's really only one. And then the last one, I think that was it, sorry. So I want to make sure we got that right. So anyway, connective tissue, I'm not going to worry about where things are located in here, but I would hope you would know adipose is also called lipid. Bone is in bone, right? The big thing about this is knowing a little bit about the matrix of connective tissue. And I would know that elastic fibers are made up of elastin protein. Reticular and collagen fibers are both made up of reticular fibers. Reticular is just thin strand or collagen, thin strands, and then collagen are bundles of the collagen fibers. You know, again, the 3 types of muscle tissue, this is a pretty important one. Again, we have skeletal, cardiac, and smooth muscle. Skeletal muscle is the only one that is voluntary. It's one of the only things we consciously control. And then cardiac and smooth are involuntary. Skeletal and cardiac are called striated, where smooth muscle is non-striated. That's why it is called smooth. In cardiac muscle are those things called intercalated discs, which are the junctions between the cells. Those are pretty big. The integumentary system, this is another one which I probably won't ask a lot, you know, all 8. I'll probably try to, you know, use 6 to 7 on this one to try and help get some things for other chapters. It is nothing in depth on here. You need to know the integumentary system is made up of 3 layers: epidermis, dermis, hypodermis. And the hypodermis is also called the subcutaneous. You need to know both of those. You need to know the epidermis and the dermis is called the cutaneous membrane, which is, scientifically speaking, the only thing that we can call the skin. So factors that determine skin color, this is melatonin, or melanin. Ooh, not melatonin. I'm getting confused here as I'm trying to make sure I get all this stuff in. But anyway, that all of us have the same number of melanocytes. but just in heredity, genetics, different people will have a different skin color because of that. Burns, I would know the 3 types of burns. Superficial partial thickness is 1st degree, deep partial thickness is 2nd degree, full thickness is 3rd degree. Superficial partial thickness barely gets into the epidermis, which isn't very deep. Deep partial thickness is going to get into the uppermost layer of the dermis. Therefore, it's going to get into the capillary beds that's under there, those capillaries, and therefore it's going to have much more reason to produce blisters. But then the deep partial thickness is going to get deep into the dermis, if not below it, and it is a very deadly, dangerous type burn. Rule of 9, remember we went over those. Factors of 9, pretty much everything's going to be some factor of 9. And so, I'm not going to go over those, but, look at your book and go through that. Arms are 9, legs 18, body, 18 in front, 18 in the back, 9 in the head. So, kind of go through those and get an idea of those. Again, the thing is, when I'm going through these, I'm not making these questions. I'm having to use the question bank from the book. So I don't have as much control, but I do have control over what is on there or not. And I will make sure that all of these questions are fair. I'm not going to give you something that kind of throws off a different curve. Now I'm hoping everybody, now we're pretty much at the 30-minute mark on this, and I hate that, but I want to make sure everybody understands and hears me when I say that this is not everything. I'm not saying that if I don't mention something in here that it is not fair game. I'm going to, everything's fair game, but I'm trying to give you a study guide to help you. Chapter 7, the skeletal system. Again, the basics of classifying bone according to shapes. Remember, there's 5 types: long bone, short bone, flat bone, sesamoid bone, and irregular bone. Long bone's femur, flat bone is the skull, short bone are carpals, irregular bone or vertebrae, and the sesamoid bone is the patella. You need to know some stuff about the microscopic structure of the long bone. You need to know about osteons and the osteonic canal. I think the book calls it the central canal, and I hate that because, again, I don't think that's a good name for it, but that is what it is. I don't know if I'm going to use anything that would use that because, anyway, I'm not going to get down that rabbit hole. You need to know how bones are formed, intermembranous, a little about it compared to endochondrial. Endochondrial is about the long bone. The long bone, the big thing about this is, it's a 2-step process in endochondrial. It starts in the medullary cavity. It starts in the diaphysis, and that is the primary ossification center, the secondary ossification center, or the epiphyses. So know basics about the difference between axillary and appendicular portions of the skeleton. Appendicular, arms and legs, and the girdle that holds it on. Axial is the head, the skull, vertebrae, and the thoracic cage. Then we get to the joints of the skeletal system. Again, this is a very basic chapter. I'm not going to get super in depth with this. You do need to know what the difference is of origin and insertion. I'm not worried about the lever stuff. You should know a little bit about how joints are classified, basic information about that. Structure of the synovial joint. This, I'm going to try to do more things about, as it says here, the pivot joint, hinge joint, ball and socket joint, instead of into the actual structures, the synovial fluid, the articular cartilage. Do expect you to know articular cartilage. Articular cartilage is hyaline cartilage. Again, hyaline cartilage is the most common cartilage. If you have a question about cartilage, guess it if you don't know the answer. Again, this chapter will be one that I'll probably be, taking a few of the questions, out of. It won't have as many, because something like the muscular system, I'm going to have a couple questions about the muscular system. So we're going to talk about the major parts of skeletal muscle fiber and functions of each. This is basically going to be talking about stuff you went over in lab. Sarcolemma is the cell membrane. It has these extensions called transverse tubules that go inside of the muscle cell. Also remember, muscle cells are called muscle fibers. That's the same meaning of both of those. In between the transverse tubule or this webbing called the sarcoplasmic reticulum. All of this is going to be around these long tubes that extend the length of the muscle fiber called myofibrils. The myofibrils are broken up into sections called sarcomeres. You don't have to worry about zones in this one or any of that, but you do need to know a little bit about actin and myosin. So inside each myofibril are these little segments called sarcomeres. They are the functional unit of the muscle. They're a contracting unit. It's made up of myosin and actin. Myosin is the thick filament. Actin is the thin filament. You do need to know Z lines are the borders. Actin is bound to the Z line on either side. Myosin's in the middle. And so when we have a muscle contraction, myosin grabs actin and pulls it towards the middle. So when we look at the actin and myosin, remember that myosin is going to have the cross bridges, the myosin heads, and then actin is going to have the binding sites. But there's going to be two big structures on actin that you need to know about. One of is called tropomin, and the other one is called tropomyosin. Tropomyosin is a long strand, and it covers the binding sites. It is held in place by tropomin. Now, the sarcoplasmic reticulum holds calcium. So when I want to cause the muscle to contract, I'm going to have a motor nerve impulse go down to the neuromuscular junction, which is the fancy word for a synapse between the nervous system and a skeletal muscle. It's going to release, when the action potential gets down to the synapse, it's going to release acetylcholine into the synaptic cleft. It's going to cause an electrical charge to be carried along the sarcoplasmic reticulum. That electrical charge goes into the cell, and it will cause a release of calcium in the sarcoplasmic reticulum. The sarcoplasmic reticulum, as it releases the calcium will bind to the tropomin. Tropomin then lets go of tropomyosin, so the binding sites are open. The myosin heads bind with the binding site, and when they do that, they have to release ADP and phosphate, causing them to change shape from pointing out to pointing in. So it's pulling the actin to the middle. And so when it's pulling the actin to the middle, it's causing the sarcomere to shorten. And since there's thousands of sarcomeres along the myofibril, it's causing the myofibril to shorten, thus causing the whole muscle to shorten. So that's how it works. Now, in order to keep this going, the ADP and phosphate get joined together to form ATP, which goes in, and the myosin heads bind to it, and as soon as they bind to it, it breaks back down, and so it goes from pointing to the middle to pointing back to the outside, and as long as the binding sites are open, it does the same thing back and forth, pulling and letting go, and so it keeps pulling that sarcomere shorter and shorter. All right. Now, in that, we have a couple energy sources. The very first one is just the ADP. Then we have creatine phosphate for longer periods of energy for a couple of minutes. But then we have to get into cellular respiration if we're doing it for long periods of time. Again, cellular respiration, just to remind you, glucose comes into the cell, two phases. Anaerobic's the first one. It consists of only one step called glycolysis. If oxygen isn't present, we can get the breakdown of glycolysis into the mitochondria. Then we have aerobic respiration, which is going to take that and go through the Krebs or citric acid cycle, and then finally the electron transport chain. Now, if oxygen is not present, then we build up something called lactic acid. Lactic acid, all of us have probably experienced this if you've exercised or done anything, work outside or whatever, and you get sore afterwards. That is the lactic acid buildup. So that is the way it works. Now, as our body goes through, we talked a little bit about oxygen debt. I don't foresee this being a question, but all it's saying is my body will let me go through activity, even in the point of saying, I'm going to give you basically a credit card, and you can run up some oxygen, but you will have to pay it back as soon as it's done. So then we go into fast and slow twitch fibers. Remember, fast twitch are the sprinters, slow twitch are the marathon runners. Fast twitch are worried about instant energy, slow twitch are worried about prolonged energy. Fast twitch are only worried about glycolysis. If I'm only worried about glycolysis, then I'm not worried about oxygen because it is anaerobic. If I'm not worried about oxygen, I do not need a lot of capillary beds. I also do not need a lot of mitochondria. We also do not need this thing that we call myoglobin, which I forgot to mention earlier. Inside muscles, we have this particular molecule called myoglobin that holds on to oxygen. The more we are using muscles, the more our body produces myoglobin. Slow twitch fibers, all right, these ones that need long periods of energy, unlike the fast twitch, which I just talked about, they will have a lot of myoglobin because in slow twitch, I desperately want to get into the mitochondria. So I'm going to have a lot of mitochondria, I'm going to have a lot of blood vessels, and I'm going to have a lot of myoglobin because I want to have prolonged energy. We've already talked a little bit about skeletal, smooth, and cardiac muscle. I'm not going to go over it again. You definitely need to know the basics of origin insertion. Nothing crazy. I'm not going to be giving you I'm not going to be getting into a lot of specific muscles, just the general idea of what origin and insertion is. The nervous system. So I've been trying to work on, got to get this, I had something fall down here. Been trying to get this where it's not taking too long, but now we're getting into some more complicated systems. But luckily, these are the ones we just got through, so hopefully they should be fresh in your mind. You know, it says first it says listing the general functions of the nervous system. This is basically going to the idea of sensory, interneuron, motor. Sensory incoming, we've got stuff that's bringing information in, the nervous system is analyzing it, sending signals out as motor neurons. So in this, the parts of the neuron, you should know dendrites are things bringing information in, the axon is what takes it away. And this is how we classify neurons based on those things coming off the cell body. So we have 3 types of neurons. This is structured, and we look at function as well, but 3 types of neurons: multipolar neurons, bipolar neurons, unipolar neurons. Multipolar neurons are by far the most common. They are basically the neurons that are going to be in the majority of our central nervous system. Most, if not all, motor neurons are going to be multipolar neurons. Bipolar neurons are very special, and I say that because they are special in the sense that they are special senses. Your taste, your sight, your hearing, your smell are all bipolar neurons. The last one are called unipolar. There's one thing that comes off the cell body. These are your normal, general sensory neurons. So those are the 3 types according to structure. The 3 types according to function are going to be sensory neurons bringing information in, interneurons, the ones that are analyzing it, and motor neurons, the ones that are sending it out. Now there are a couple of other cells called neuroglial cells that help neurons. Neuroglial cells, there's going to be a different set in the central nervous system versus a different set that's in the peripheral nervous system. 2 of them are designed to provide myelin on the axon. In the peripheral nervous system, they're Swan cells. That's what I would kind of concentrate on. Most of the things we talk about as far as myelination, we're going to talk about the Swan cells. But do not overlook the fact that in the central nervous system, the cell that provides myelination is called an oligodendrocyte. All right, so that's an important thing to know. Now, in the central nervous system, we also have things called astrocytes and microglial cells. Astrocytes and microglial cells are ones that are worried about keeping the area around the neurons in proper order. Astrocytes are going to look like stars. They're going to basically be the butlers. They're keeping sure everything is in its proper order and all good. Microglial cells, I would associate them with phagocytic properties because they're more the janitors. They're going through getting rid of things. And then finally, the epidemial cells. Anytime you hear epidemial cells, you should automatically think cerebral spinal fluid because that's their job. They have little cilia on it that cause the cerebral spinal fluid to flow through. The only in the peripheral nervous system, the only one that we have there beside the Swan cell is what are called satellite cells. And satellite cells do the job of astrocytes and microglial cells. All right. Again, the big one is the Swan cell. Now, cell membrane potential and events leading to the generation of an action potential. Remember that in a neuron. Let's see if this works. I don't want all that stuff. In a neuron, I just want to write. The heck? All right. I shouldn't have even messed with that. I need to get out of this and go back to what I was doing. So anyway, if I have a neuron, why did I try that? Why did I try that, people? I was doing so well. So if I have a neuron and I'm going through and I am going to be, I'm so sorry. I'm so sorry. Now I don't even know how to get back to this. Oh, this is, there we go. Back to my thing. So if I've got a neuron, there's a dividing line. The axon comes off the cell body at a place called at the axonal hillock. There's a change. Everything from the cell body and dendrites on that one side of the axonal hillock has what we call graded potentials. That means that there's all kinds of different things coming in. Nothing is organized, nothing is set, nothing's cookie cutter. I'm going to have some things that are stimulatory, which are going to make it easier to send a signal. Some things are inhibitory and makes it harder. But at that point of the axonal hillock, then we have what's called an action potential. What goes down the axon is absolutely the same every time. All right. So the big thing is to understand we have two major ions. There's three overall, but two major ions. Potassium is on the inside of the cell. Sodium is on the outside of the cell. When I have things disrupt the cell membrane potential, which is the charge, the difference between those, it will cause disruption. So if sodium starts coming in, then I'm going to have a problem with my normal resting membrane potential, which is registered as a negative 70 millivolts. But the dance between sodium and potassium is what causes all of these things to go on. So if all of that summates, all of the graded potentials reach a certain point, which is negative 55, so to go from negative 70 to negative 55 is positive 15 millivolts. So if the graded potentials sum together to get a positive 15 difference at that axonal hillock, the gates then open, and I'm going to have a very organized wave that's going to go down to the synapse. Now, that is called an action potential. The ion channels from the axonal hillock down to the synaptic bulb are going to be voltage controlled. They're going to be controlled by the change in ions. So negative 55 is when the sodium channels open. So sodium rushes in, and I'm going to have a quick depolarization of the cell membrane. That means that all of a sudden it becomes positive. All right? So negative 70 goes all the way up to a positive 30. Now in that little area, I've got two positive ions. Just like two positive magnets, they do not like each other. And so at positive 30, the potassium channels open and potassium gets pushed out. Now I'm going to have a repolarization, but I'm going to have a repolarization at the cost of the normal setup of ions. So I'm always going to be pushing forward towards the synapse because behind me, the ions are messed up. So this is again how the action potential moves down the axon. And it's going to jump from node to node. If I have Swan cells on there, or if there's oligodendrocytes in the central nervous system, there's going to be little areas of exposed axon between those cells, and it's called the node of Ranvier. And so the little, it's going to jump from node to node as it goes through, and that's called saltatory conduction. Now, identify changes in membrane associated with excitatory and inhibitory neurotransmitters. All right, so as the neurotransmitters come in to the graded side, you know, they're either going to make the postsynaptic, as it says, cell more likely to send a signal or less likely. If it's more likely, it's called excitatory. If it's less likely, it's called inhibitory. All right. Now, chapter 11 is when we get into some more in-depth stuff about the central nervous system. Now, chapter 11 starts with the meninges, and it talks about this little section of membranes that basically quarantine, what a weird word to use now, quarantine my central nervous system from the rest of my body. And there's 3 of them. Right, basic information here. That's all I'm looking for. Basic information. So the outermost layer is called the dura mater. The middle layer is called the arachnoid matter. The inner layer is called the pia mater. The pia mater would be considered the visceral membrane because it is what covers the brain and spinal cord. It is the skin of it. And so the dura mater, arachnoid mater, pia mater. Now, in the skull, the dura mater is pretty much attached to the periosteum of the inside of the cranial cavity. Once it gets into the spinal cord, there is a space between the dura mater and the bone that makes up that vertebral foramen. That space is called the epidural space. Epi means above, so it's just telling you that. Then there's a space between the arachnoid matter and the pia mater, and it's called the subarachnoid space. And this is where you're going to find cerebral spinal fluid. Now, cerebral spinal fluid, the big thing is know that it's made in a set of capillaries called the choroid plexus, a special type of capillary that releases this. And this is where you're going to find those epidemial cells that are going to be, the cilia is going to be beating back and forth, causing a flow. Trying to find the major parts of the brain. All right, so the brain, brainstem, and spinal cord. Remember, so this is, I'm going to stick with the basics on here. Now, some of this you'll see isn't necessarily basic, like the basal nuclei, but you should know the brain. The brain is made up of 2 hemispheres. It's made up of 4, and there's 5 lobes. I'm only going to hold you responsible for 4: frontal, parietal, temporal, occipital. The band of tissue that connects the 2 hemispheres is called the corpus callosum. The corpus callosum is always, for me, I always describe it as a sideways ear because that's what it looks like. It allows the 2 hemispheres to talk together. Underneath the corpus callosum is an area called the diencephalon that has 2 things you learned in lab, thalamus and hypothalamus, and I'm going to come back to those. Then you have the brainstem. The brainstem has the midbrain, the pons, and the medulla oblongata. Again, we'll talk a little bit about those when we get a little more specific. But then the last part is the cerebellum, the lobe that's in the back. This little cerebellum means little brain. So when we start looking at that, right, the big thing is first the diencephalon, the thalamus, the hypothalamus. Remember, the thalamus is all about sensory information. It is organizing all the information coming into the cranial cavity and routing it to the portions of the brain that need to hear it, which is its counterpart. The basal nuclei, you need to know, is doing the same for motor impulses. As the motor impulses are trying to leave the brain, it goes to the basal nuclei and it is smoothed out. The hypothalamus is the crossover between the nervous system and the endocrine system. It controls the pituitary gland. So it is a part of the, it is the go-between, and it is a big part of homeostasis. It is part of homeostasis because it controls the endocrine system. Now on the brain stem, that first part, the entryway, is called the medulla oblongata. It's the other big part of homeostasis, and it is directly nervous system control. It is a place of big reflexes for things like the heart and the lungs and the blood vessels. So the thalamus and the basal nuclei are kind of two parts, the yin and the yang, so to speak, two opposite sides of things. I'm not worried about the pons in the midbrain right now. I want you to know that the cerebrum is about coordinating the sensory and motor impulses to help make sure muscles are, move smoothly. Again, this could be a whole chapter in and of itself. It would be very basic on this. Hemisphere dominance, know that in most people, the left brain is dominant, the right brain is not. And so there are a few people that are opposite in that, but know that the left brain is more analytical, less emotional. The right brain is more emotional. Reflex and the behavior, I would know the 5 parts of a reflex arc. It would be one question. I probably have it, I think I've seen it on there where they put them in order. Know that it's receptor, sensory, which is also called afferent, interneuron, which is the only part that's in the central nervous system, then motor or efferent, and effector. That's the reflex arc. I wouldn't get too crazy about knowing the tracks, but I would know, to me, if I was a student, once you realize how simple that is, I'd kind of want them to be. You should know that all ascending tracks are motor, all descending tracks are sensory. If you see a track that is named, which I doubt that I'm going to do, but I'm just going to, it's fair game. But if I label a tract and it says it has the word spine at the front, that means it's going from the spine up to the brain. That means it's motor. If it has a part of some word with and ends with spine, it means it's going from the brain down to the spine. So it is descending and sensory. All right. Now, the big one is this, because this is probably the most useful portion of this class going on, which there's plenty, obviously, all of this builds as you go through. But knowing the sympathetic versus parasympathetic division of the autonomic nervous system is huge. So, autonomic nervous system. Autonomic nervous system is the automatic pilot, right? So we have this autonomic nervous system, which is controlling things, underneath our conscious control. There's 2 portions to it, parasympathetic and sympathetic. Parasympathetic is normal operating procedure. Sympathetic is under stress or during activities. Sympathetic is all about using energy that we've stored. Parasympathetic is all about storing energy from what we've just eaten. Now, sympathetic, fight or flight, nervous, everyone should know that that's associated with adrenaline. As long as you can remember adrenaline is the same thing as epinephrine, that should tie everything together. Now, there are 2 types of epinephrine, norepinephrine and epinephrine, just like there's 2 types of adrenaline, noradrenaline and adrenaline. I am not going to try to trick you on either one of those. At this point, this is above our pay grade to know the difference of those. If you see either one of them in a question, because it's going to be from a test bank, know that I am not trying to, I'm not going to put norepinephrine if epinephrine's the answer. Right? You know what I'm saying? So I don't want you to think that you get tricked on it. Think of both of those as the same thing. All 4 of them basically is the same thing. That should always be associated with the sympathetic nervous system. So when we looked at these, we talked about how there is a difference in the makeup of the neurons coming out of the central nervous system, because in the autonomic nervous system, it's a relay race. There are 2 neurons. I'm going to have a pre-synaptic or pre-ganglionic neuron and a post-synaptic or post-ganglionic neuron. So in the parasympathetic nervous system, that first neuron, the pre, it goes all the way out to the target organ, and then the synapse is pretty much going to be right there. So if I'm going out, if I'm sending right now, I'm not stressed, I don't think. My body's sending a signal out to my heart. That neuron, that first neuron, the presynaptic neuron's going all the way out, gets out to my heart, and is going to synapse with another neuron that's basically right on my heart. That second, that postsynaptic neuron is very short. goes right to it, boom. Now, in the parasympathetic nervous system, both of those neurons release acetylcholine, most common one. So they're both called cholinergic neurons. Cholinergic means acetylcholine. In the sympathetic nervous system, the first neuron is very short because it wants to synapse really quick and spread that message out like it's an Amber Alert. So it's going to barely get outside of the safety of the spinal column, and it's going to synapse, and it's going to spread all out. Now, that first synapse still uses acetylcholine. So the preganglionic or presynaptic neuron in the sympathetic nervous system is still cholinergic. It is that second neuron that is going to release epinephrine, norepinephrine, or adrenaline to the organs. And so that second or postsynaptic, postganglionic neuron is the one considered adrenergic, which means it's adrenaline. And it should make sense. Look, if I've got the heart and I've got two things coming together to talk to the heart, one of them causes it to beat harder, boom, boom, boom. The other one causes it to beat slow. I need to have two different messages that get there. So it has to have two different neurotransmitters. One of them tells it to beat slow, one of them tells it to beat fast. And so there's, of the four, if we look at the two systems, sympathetic and parasympathetic, there are four neurons there, two preganglionic, two postganglionic. The only one that is not releasing acetylcholine is the postganglionic neuron in the sympathetic nervous system. And I hope that makes sense. Again, we're right at an hour and I'm trying to wrap this up. Now, on this, chapter 12, we went in lab, you should have went over the 5 types of receptors. You should know them. Chemoreceptor, chemicals. Thermoreceptor, temperature. Mechanoreceptor is about pressure and mechanical changes, stretch, tension. Photoreceptors, which are about light energy. And then finally, nociceptors, which are pain. Now, you went over the ear and the eye in lab. The ear uses mechanoreceptors. You should know that. It is pressure waves in the air that go through here. They hit that tympanic membrane that gets transmitted as neural impulses. It's pressure waves. In the eye, it's photoreceptors. You should know that, all right? So the basics about those, but definitely those two in particular, mechanoreceptors and photoreceptors, knowing that the ear and the eye, which one uses which. I do want you to know the difference between sensation and perception. Sensation and perception. You have an unbelievable amount of information coming into your brain right now. So much stuff is going into your brain, and most of it you're not aware of. All of it is classified as sensations. The things that you're aware of, those sensations that you're aware of, like let's say, you know, I take a pencil that's got a sharp thing and I don't realize and I stab myself with it, poke it. That is now a perception because that sensation I am now aware of, consciously aware of. So it's a perception. I hope that makes sense. Now, our body is constantly adapting to our surroundings, so we go through something called sensory adaptation, which is simply that as we go through life, your sense of smell is the easiest one, which is a chemoreceptor. If I go, you know, we've been cooped up, so let's say I leave my house like today, and I go back, I'll probably smell what my house smells like. Hopefully it's good. I think it's good. I mean, it's been clean. It's clean all the time, you know, whatever. But let's say I left dirty dishes out, and, you know, dirty clothes. There's a whole commercial now about going nose blind. That's sensory adaptation, all right? I'm not going to get into dermatones, referred pain, which is really pretty important to understand that, you know, your body's going to send out signals called referred pain because your organs don't have a direct connection to give you a perception of it hurting. So it'll bypass that and use one that you do have. So for instance, heart issues, first sign is arm pain, right? Arm pain and jaw pain. So it's not, your arm's not, your arm and your heart don't have anything in common, but it's an area where both of those neurons come together in the central nervous system. And so the heart, if it's hurting, it can't directly talk to the brain. So it synapses here, and then our body will feel it like it's coming from the arm. That's how we get referred pain. I would know the basics of the ear, and I would definitely know what cranial nerve works the ear. Cranial nerve 8, the vestibulocochlear nerve, and it is a sensory-only nerve. You don't have to worry about the auditory pathway, but you should know auricle or pinna. And auricle is probably the one that's going to use. You'll talk about auricles when we get into the heart in 211. External acoustic meatus, tympanic membrane, is what we commonly call the eardrum. You need to know that. Your sense of hearing is going to be found in the cochlea. Your sense of balance is going to be found in the semicircular canal, stuff like that. When we get to the eye, again, very basic stuff. You should know that the eye has 3 layers. The outer layer consists of the sclera and the cornea. Sclera is simply this thickened white part. The cornea is the clear part of it. Behind that, we're going to have the choroid coat. And the choroid coat is going to mainly be vascular. You don't have to know that. But the choroid coat is going to have some pretty important structures that are behind the cornea. You're going to have first the iris. You need to know the iris is a round, smooth muscle. Inside the iris, there's a hollow circular spot called the pupil. The pupil and the iris are designed to regulate the amount of light that enters your eye. Behind the eye, there is a thing called the lens. It's designed to focus the light. It is held in place by suspensory ligaments and a thing called the ciliary body. The ciliary body is a muscle that's going to pull on the lens to flatten it or let it relax. To be honest with you, I doubt that if this is probably only going to be about 6 questions on here. I don't think I'm going to get into any complicated stuff on there. But you do need to know that the lens represents a dividing line in the eye. Posterior cavity is behind it, filled with vitreous humor. Anterior cavity is in front of it, filled with aqueous humor. The posterior cavity is going to have an inner lining that we call the retina. The retina is made up of rods and cones. You do need to know they're photoreceptors. You need to know that rods are for low light image, and it'll produce a black and white image in your brain, and it will be very grainy. Whereas cones produce a color image. Now there's 3 types of cones, and we're not going to get into it in here right now because of the fact that just the way things are. But just know that cones produce color images, and they require much more light. Now, in the retina, there is a little area called the fovea centralis directly behind the pupil that is entirely cones, no rods. Now, again, if you've ever, I don't know how many people like to look at the stars. And there's actually right now, I don't know if you've heard about this, I think it's called Skylink or Starlink. It's a group of about 60 satellites that goes back and forth. I think tonight, I should look on, I think it's tonight on at 9. It might be tomorrow. Anyway, it's going to go overhead. It's really cool to see. But if you're out looking at the stars and there's a dim star, if you look directly at it, a lot of times you won't be able to see it. But if you look off to the side, it'll kind of show up. And all that's doing is, it's because if you're looking directly at it, if it doesn't have enough light energy to cause the cones to fire and send a signal, if you look a little bit away from it, the rods are there and it will help send a signal. So if you've ever looked up at the night sky and wondered why certain dim stars you can't look directly at, that's why. So that's the general stuff. Again, I'm not saying that this is all that's there. I'm just trying to give you a quick review. I hope everybody's doing well. Again, like I said at the beginning, the test, I'm going to open it Wednesday morning. It'll be open through Thursday night. I'm only doing that so that we can have some time if something comes up. I've got Friday. As a matter of fact, I'll go ahead and tell you, and I'll send this in the email. I'll open it up till Friday. That gives me the weekend, but if something happens, all grades are due on Monday, and so it's going to be final. So, I hope everybody's doing well. I hope that this helped. I wish that I could do more. You know, you're going to be fine. You know, just trust yourself. All right. Take care, everyone, and be safe. And I look forward to talking to you all soon.